Hi guys, David here with another science update. So today we're going to learn about how mussels are inspiring new generations of adhesives for dental and medical applications. So to start off, we're going to look at an article that actually came out this week called How Mussels Make a Powerful Underwater Glue. So the article says that blue mussels spend their days being buffeted by crashing waves. They manage to stay tethered to the rocks or their fellow mussels thanks to a highly effective underwater glue they produce. Because achieving adhesion in the presence of water is so challenging, scientists who are interested in producing effective adhesives for use in wet environments, for instance surgical or dental treatments, have turned to mussels for inspiration. In a paper published today in Science, a McGill-led international research team reports that after a decade of work in the area, it has succeeded in uncovering the cellular mechanisms by which the mussels fabricate underwater adhesives. So before we dive into that, we should just look at what a mussel looks like. So we can see here in this image that we have the main body of the mussel, and then we have something called the mussel foot. The mussel foot is responsible for generating the glue or the adhesive, and the adhesive the adhesive structure consists of a Bissell thread, which you can see here, this kind of like white line, and then it has what they're describing as a disc-like feature. I don't really see it. It doesn't look like a disc in this picture, but I'll take their word for it. And apparently this disc-like structure is called a plaque, and that's where the adhesive action takes place. So moving on, the article says, by gathering information at a subcellular level, the researchers discovered that within the mussel foot, there are micron-sized channels. So this is saying ranging in diameter from about 1/10 to the full width of a human hair, so very, very small channels, which funnel the substances that come together to make the glue. And there's two main classes of substances. One are these condensed fluid proteins, which are made in tiny sacs or vesicles. And then the other substance are metal ions, including iron and vanadium, which were taken up from the seawater. The metal ions, which are also stored in small vesicles, are slowly released in a carefully timed process. And then when the two compounds come together, the condensed fluid protein and the metals, they sort of cure to actually form the strong adhesive. So in the first part of this video, you can see the Bissell threads are being produced by the mussel. And what we'll see soon is actually a CT scan of what it would look like inside the mussel foot. This is the specialized organ that makes the glues, and these little blue channels are basically what we were talking about. These are these little channels where the fluid-like proteins are going to be put through, and then the green part is the regions of the mussel foot where the proteins for the glue are produced and stored. So they're produced and in these green areas, and then they're funneled out through these blue areas where they can mix with the metals to cure. Very cool. What they say is the accumulation and use of vanadium, so it says it's using the metals are vanadium and iron, is interesting since only a few other organisms are known to hyperaccumulate vanadium. The researchers believe that it plays an important role in hardening the glue and are continuing to work in this area. So I think the main findings from this new research are really this idea that there is this structure in the mussel foot which is actually combining the proteins and the metals to produce this curing. And then the other thing is the fact that they found that it was iron and vanadium that are responsible for curing these proteins. And by curing, I just mean hardening. So I wanted to do a little bit more reading about the topic, and I found that in research that people have done to actually make biomimetic glues, so they're trying to make glues which are inspired by the mussel glues, and to do that they actually did quite a bit of work in characterizing the proteins that go into the mussel glue. In particular, they found that a lot of the adhesive action comes from proteins that have an amino acid called L-DOPA. And you can see here this structure is basically a 6-membered carbon ring that has 2 OH groups or 2 alcohol groups. That's the main feature. The rest of this molecule on the right, this NH2 and the rest of it, would be part of the backbone of a protein. So this is being the side group is the more important functional group when it actually comes to the adhesive action. And so when researchers make polymers that are trying to mimic this, so we hear this would be the chemical structure of the protein for the mussel, and then this would be the chemical structure of a synthetic glue, which is trying to mimic it. They're going to make structures like this that have this, again, the 6-membered carbon ring with the 2 hydroxyl groups. And that's called a catechol group. And so if you look into research, this is a review article about recent progress of mussel-inspired underwater adhesives. You can see that a lot of different research groups have undertaken to make many different chemical structures of artificial or biomimetic glues, which are trying to mimic what's going on in mussel adhesives. And they have all kinds of different structures, but you can see in red here, this same motif, this catechol group, the 6-membered ring with the 2 OH groups shows up everywhere. So that's kind of really the main thing. But what's interesting about this article here, which is from 2017, is that in this article, there's no mention of vanadium or iron. And so this idea that it's vanadium and iron that are actually causing the curing may be a new insight, which could hopefully take this biomimetic polymer development to the next level. So you can see here, for instance, they're looking at a structure of how the metal coordination actually works, or how the glue actually works, is they have these little M's which stand for metal oxide. And the idea is the catechol group is going to coordinate with metal oxides. So they understood that much, but as I said, in this paper there's no mention of vanadium. So particularly, I think that this new insight that came out this week about vanadium being a key element of the whole glue hardening process for these underwater adhesives may have some impact in developing an even more improved version of the biomimetic polymers. And when I say biomimetic, I should specify that the bio is short for biological, which means pertaining to a living system, and then the mimetic part means mimicking. So a biomimetic material is something that is mimicking something found in a living system. But anyway, I thought this was a really interesting new finding, especially essentially the intersection of how they used cell biology and connected that to the polymer science that people are already doing. And the hope is that by combining these insights, they may be able to, not too long from now, make even more improved versions of synthetic glues that could work for next generation medical glues and also for dental applications. Well, thanks so much for watching. This has been another Science Update. If you liked the video, please go ahead and like and subscribe, and I hope you'll join me again soon in the future. Bye.